The main objective of this lab is the synthesis of tris-1 10-phenanthrene iron 2 chloride, which creates a deep red crystal. After the lab, we will calculate the percent yield to ensure we've properly synthesized the red crystals. The first part of this lab is called the synthesis, where we will combine ferrous ammonium sulfate and Fen in order to prepare our solution. Before performing the lab, we first need to wear the appropriate attire required, which includes safety goggles, an apron, gloves, and closed-toe shoes. First, we need to weigh 0.86 grams of ferrous ammonium sulfate and 1 gram of Fen on a weighing paper. Then, into a 50 milliliter beaker, we will use 10 milliliters of 1 molar HCl and use the 0.86 grams of ferrous ammonium sulfate that we weighed in order to completely dissolve the ferrous ammonium sulfate. Now, into a 150 milliliter beaker, we will dissolve the 1 gram of Fen by using 10 milliliters of methanol. These are what the two mixtures should look like after they are completely dissolved. On the left we have the Fen, and on the right we have the ferrous ammonium sulfate. Now, using the two mixtures, pour the dissolved ferrous ammonium sulfate into the dissolved Fen and stir. As you pour and mix the two solutions, you should instantly see a deep red color change starting to occur. Our second part of the lab is called the workup. In this part, students who aren't careful tend to lose product, causing a lower yield. So being patient is the key to getting the red crystals that we need, as we will see later in the lab. The reason we need to be patient here is to make sure we have the crystals needed in order to analyze them in the next lab. Weigh 2.5 grams of NaCl and dissolve it with 10 milliliters of water into a small beaker. With the dissolved NaCl, add it to our Fen solution and stir. With our new solution, we will set up a gravity filtration to remove any unwanted solids before we precipitate it. Shown in the video is me setting up the gravity filtration. You will need a clamp for the funnel, filter paper, and a beaker to catch all the waste. First, you need to wet the filter paper with a little bit of DI water. Then you need to pour the solution and wait for it to be completely filtered. After the solution has been completely filtered, you want to use DI water to rinse off any red solution that is in the beaker or that remains in the filter paper. And remember, the more water you use, the longer it will take to completely filter. Now, we need to add 2 to 3 boiling chips into our new solution and put the solution on the hot plate. Before we begin boiling the solution, we need to ensure that we add the watch glass properly on the beaker. We want to add a glass rod in the beaker to ensure that we create an opening for all the gas to escape. As shown without the glass rod, all the condensation that occurs on top of the watch glass will just continue to drip into the solution, causing no reduction to occur. While we wait for the solution to boil down to about 1/3, add 5 milliliters of water, 5 milliliters of methanol, and 10 milliliters of acetone, each into a separate beaker, into an ice bath. Now when the solution is done boiling, we will use tongs to remove it from the hot plate and let it cool on the bench for about 10 minutes before adding it to the ice bath for additional 15 minutes. The act of cooling the solution helps the crystals form. This is what my solution looked like after 20 minutes of cooling. Each solution is different with how long it will take to form the crystals, so ensuring that your solution has cooled for about 20 minutes will help yield better results. Now we are going to use a vacuum filtration in order to extract the crystals that we let cool down and form. In a gravity filtration, we want to make sure that the shorter pipe inside the bottle is going to be connected into the vacuum, and the longer pipe inside the bottle is going to be connected to the flask. Using the water that we kept in the ice bath, pour it into the solution in order to help extract out the crystals that are formed on the beaker. Now using a little bit of methanol, we will wet the filter paper. After we add our crystals into the vacuum, we will wash them with ice cold methanol, ensuring that we spread the methanol across the crystals. After we wash the crystals with methanol, we will then wash the crystals twice using the ice cold acetone. After leaving the vacuum on to let the crystals dry, we will take both the filter paper and the crystals and put them on a petri dish to where we will then put them in the oven. After the crystals are done drying, add them into a vial for the next lab. Your crystals should more or less look like this. Before finishing the lab, make sure to transfer any filtrates or washes to the heavy metal waste container. Now I wanted to show you what would happen whenever we use too much methanol in this experiment. Right here I have some red crystals and some room temperature methanol, and as I pour this methanol in the red crystals, it's going to quickly dissolve them, showing their effectiveness in this lab. And as I pour this out into another beaker to show y'all how effective it is, we could almost see that the crystals have almost completely dissolved. And then after a couple of seconds, it's pretty cool seeing how they just completely disappear due to the methanol. And finally, after you've recorded all your data and observations, we will now be able to calculate the percent yield for our synthesis. Thank you for watching the video, and I hope it helped.